Welcome to the second video of this series or for this last unit in our class. And if the environmental noise is too much or my daughter's seen through the back, I apologize for that. But this is the unit getting into the chemistry that explains how the drug actually works or how different drugs work. The sort of explaining the rationale behind the drug design and the things that we'll talk about here in this video and maybe the next because um, just because of the limitation of my laptop and whatnot, I think 10-15 minutes is kind of the max of each video that I could make. So anyway, I have to break this up into quite a few. Sorry about that, but the implication is going to be both on the pharmacodynamic as well as pharmacokinetic properties of drugs. And hopefully those words make sense to you. Okay. And I think so far in this unit, in this chapter, we have discussed what pharmacodynamics mean and pharmacokinetics mean in the in-person class, I think last week. And then today, I basically mainly want to talk about, well, in this video anyway, sort of the rationale behind the design of anti-cancer drugs. Then I think in the next video, we'll talk about receptor-based drugs and whatnot. So that's the plan. And there's different types of anti-cancer drugs. That's not really like one category, at least as far as the chemical mechanism goes. These are some different categories you could think of. Alkylating type of agents being used as anti-cancer drugs. There's a class called anti-metabolites. We'll talk about those. And a lot of these are natural products to begin with anyway, but there's a specific class of compounds that, for whatever reason, oncologists, doctors who treat cancer, tend to call natural products. So that's, to me, kind of doesn't make sense because there's a lot of anti-metabolites that are based on natural products and protein kinase inhibitors that are based on natural products. Anyway, there's a class that pharmaceutical and medical physicians tend to call natural products, and we'll explain what they are. And I hope what it makes sense to you when we say kinase inhibitors. But if you recall why kinase is important, because they tend to turn on and off different types of enzymes in different systems within biology, physiology, right? And we'll talk about like why those things make it for good drug targets. And we won't have time to talk about organometallic compounds being anti-cancer, but some of the earliest cancer drugs are actually not even organometallic, but metallic compounds like cisplatin, which is a DNA cross-linker that's based on platinum. We also won't have time to talk about hormones being type of cancer drugs, but and then we also want to talk about angiogenesis inhibitors, mainly because that used to be kind of a major thing maybe 10, 15 years ago. But in the last 10 years or so, we've learned that the angiogenesis inhibitors actually have sort of the opposite effect on cancer. The word angiogenesis means the, the process of generating new blood vessels, and rationally, it seems to make sense that cancer tumors, they tend to have to produce a lot of new blood vessels for rapid growth, and so therefore maybe the compounds that would shut that process down would help stop growing the growth of cancer, and that was a rationale that made sense to us, of course, until 10 years ago. But angiogenesis inhibitors, once we put those things into clinical trials, we learned that cancers actually tend to come back with vengeance, and so that is kind of an outdated mode of treating cancer today. And so these are some different types of cancer drugs and their mechanisms of action. Um, there's some cytotoxic agents that just are good at killing cells, whether it induces apoptosis, that's a programmed cell death, um, uh, or there's also agents that stop the process of uh, growth of microtubules or recycling of microtubules. Those are the protein ropes, so to speak, that get formed inside a cell that um, are used for transportation of different types of macromolecules. Usually proteins are transported from like one place to another using microtubules, but also the genetic materials like DNA as well. And the most famous example, you might have seen cartoons or microscope pictures of cells undergoing mitosis or cell division, and then you see all these like spaghetti like hair-looking structures like pulling the chromosomes apart into the two poles during cell division or mitosis, and that hair structure is all microtubules. And then there's also topoisomerase inhibitors. That's another molecular target within a cell that's pretty active in cancer. And those are the enzymes that unwind the chromosomes so that DNAs can be read for transcription and then later on translation needed for cell replication. Right? And then there's another class of compounds, as we mentioned, called antimetabolites, and that shuts down the metabolism, or especially anabolism, or biosynthesis of molecules, specifically new DNA molecules. And the rationale, of course, is that in adults, only the cancer cells or there's not very many rapidly growing tissues in adults, and so for that reason, it's usually considered safe to shut down new DNA molecule biosynthesis. And then, like we were saying, anti-angiogenic compounds that sort of got phased out of favor, mainly because of the finding that cancers tend to come back from this with vengeance. Initially, you can stop their growth, but it's not a good idea for treating cancer, which is kind of a unfortunate turn of events. My PhD study actually centered around anti-angiogenic compounds, but that's an outdated idea today. But let's go back to the first thing we mentioned, and that is the alkylated type of compounds. And we learned a lot from actually what we call mustard compounds, which we'll talk about that in a second, which was unfortunately used a lot as a chemical weapon uh, in World War One, and uh, this is like before Geneva Convention and whatnot, and there's actually quite a bit of horrific and unethical human experiments that were done back then. But actually, today we know that mustard or alkylating agents actually work against cancer because of some of those human experiments, ironically. And the way that works um, is by a series of SN2 reactions. And these, to distinguish them from the chemical weapon type of mustard compounds, we call these nitrogen mustards. And that's not really because of their appearance, like they look like mustard, um, like yellow gas or anything like that. In fact, nitrogen mustards usually are not gaseous, either liquid or solid. And under the structure is set up such that it's set up for SN2, intramolecular SN2 reaction to kick off the chloride, making this three-membered ring or aziridium ion. And of course, this is not super stable because it's a cation. It's charged and susceptible to ring opening again by another nitrogenous nucleophile, like the basic nitrogens in the nucleobases within DNA. And once that happens, of course, the DNA can get alkylated like shown here. And of course, at this point, maybe you could tell where we're going with this. On the second half of this molecule, you could go through the same thing where you undergo intermolecular ascent tube, form the three-membered ring, which could be opened up again by a DNA nitrogenous base ring opening. And what this results in is, of course, sort of a random cross-linking of DNA strands, thereby sort of disabling this DNA from being ever used again. This is the type of bond that you would not see in nature. And there's some modification of DNA that's sort of natural, and our enzymes can handle that, but this is unnatural modification of DNA that no human enzyme knows what to do with, and so this DNA essentially gets permanently disabled. And of course, that can become advantageous in rapidly growing cancer tissues that are undergoing rapid replication. So there's no more replication, no more growth, and that's good for treating cancer. But as we said, that this idea concept was mirrored or copied from the original mustard gas design. The original mustard gas that was used in the chemical weapon has this sulfur nucleophile, and this is actually somewhat volatile. And then, so you guys probably have heard of the mustard gas. That's the chemical of mass destruction, weapon of mass destruction type of chemical weapon used, unfortunately, in World War One. And so today, here's an example of some fairly commonly prescribed mustard, nitrogen mustard agents, especially cyclophosphamide is a really well prescribed drug. I bet you probably know some relative who's been prescribed this chemotherapy somewhere. And it's an interesting drug because it's actually a pro-drug, and that's a word that you might want to be aware of. That it has nothing to do with whether you're for or against, like uh, pro-life, pro-choice, that kind of a meaning. But it just means the word pro in this context means like before, prior. So this is the structure that is prior to the active form. And the idea is this compound gets metabolized in your body in a predictable way, so that it's actually the metabolized version that is active. And can you guess as to what kind of enzyme could do this transformation where you introduce an alcohol on an alkane? And if you guess cytochrome P450, you would be correct. And of course, that introduces that amino functional group, which is susceptible to hydrolysis ring opening. And then once that ring opens, this phosphate ester can be fairly easily hydrolyzed. And this is the actual active form. And of course, we see the nitrogen mustard portion that undergoes cross-linking of DNA. Now here's another interesting nitrogen mustard drug where we see the what we might call the pharmacophore or the active end of the molecule. This is where the action happens. And then on the other side, you see this steroidal skeleton. This is often used in like prostate cancer and stuff like that. But the idea is this estrogen looking like steroidal looking portion it guides the molecule where it should go, and that's the function of the right side of the molecule. And then the left side of the molecule actually does the disabling of DNA, thereby killing the cell. So one of the goals for this unit or for this pharmaceutical chemistry part of the chapter is that some of the drugs you can kind of tell uh, what the mechanism of action is based on the structure. And for those compounds, especially, I want you to be able to sort of not guess, but make an educated guess about what the mechanism of action is based on the structure. Like, for instance, if you saw estromustine, right, like you could tell that it's a nitrogen mustard alkylating agent. And Here's another class of an anti-cancer drug, anti-metabolites, and both of these, maybe without uh, me sharing what the mechanism of action is, you might be able to guess what that might entail, especially if you think about all the primary metabolite structures, right? Like, what do these look like? They, of course, look like this one, pentostatin, looks exactly like a nucleotide or nucleoside, and then fluorouracil obviously is mimicking uracil or thymine type of nucleobase structure. And so, uh, anyway, maybe you could guess where we're going with these. These both inhibit DNA synthesis, um, and most antimetabolites um, are used as either anti-cancer or antiviral drugs. And you might see the reason why as we think about the mechanism of action. So this is a normal metabolism that happens. What I'm showing you on this slide. Uh, this is the nucleoside that would become, the A's in the DNA, adenosine, the deoxyadenosine. So this is part of DNA, not RNA. And when deoxyadenosine accumulates, that signals to our cells that we should uh, uh, increase the um, production of deoxy-ATP. And then, of course, this is different than ATP because it's lacking that SET2 hydroxy group. It's based on the DNA deoxyribose scaffold, not the ribose for the RNA. And this DATP is a potent inhibitor of ribonucleotide reductase. That's enzyme that converts ribose. Remember, that's used for RNA synthesis. And it reduces ribose to deoxyribose, which of course is the building block for synthesizing DNA, which happens a lot in a growing cell, right? And the reason why your body may want to produce DATP, a potent inhibitor of that enzyme required for biosynthesis of deoxyribose, of course, is if this is accumulated, that's a good indication that you're just doing way too much DNA synthesis in your cell. So that's sort of the feedback loop mechanism that regulates all these DNA synthesis and whatnot in a healthy cell. Okay, but of course, some of this regulation is out of whack in a cancer cell, because in a cancer cell, you want to create way more DNA than is necessary. Same with a virally infected cell. So, what this antimetabolite does is it's to mimic the transition state of this enzyme that regulates the production of deoxyadenosine, or I should say the quantity of deoxyadenosine. And the way that happens, you might recall talking about this back when we talked about the natural degradation mechanism that happens on nucleobases. They can get hydrolyzed going through aminal intermediate, and then it can become deoxyinosine. Right? This is not any of the four nucleosides in DNA, and so this becomes other things in our body, so thereby reducing the amount of DNA biosynthesis. And this is another mechanism that our body copes with, or regulate, I should say, regulate um, DNA quantity. But like we said, like these systems like purine deaminase regulating mechanisms goes out of whack in a cancer cell. And one of the things that this drug pentostatin does is it looks just like this transition state in hydrolyzing the adenosine nucleobase, right? And so this enzyme would tightly bind to this compound, because remember how enzymes work? They usually have the active site looks usually not quite like the substrate and not quite like the product, but looks more like the transition state, and thereby sort of stabilizing the transition state, right? If you think about the reaction energy diagram, if the transition state can be stabilized, then the activation energy gets lowered, and so therefore the reaction happens a lot more quickly. That's how enzymes catalyze a reaction. And so if you make a transition state mimic like this that looks like the transition state, that lowers the transition activation energy for this deamination reaction. And that's how this inhibitor is able to inhibit purine deaminase, which means this compound accumulates, which means that the ATP accumulates, and that'll shut down the overall biosynthesis of DNA. That's the idea. Okay, I hope that makes sense. That's one way that antimetabolite works. And then the last antimetabolite example we might talk about is the oops, the oh no, with the fluorouracil, but my PowerPoint's not doing well, and I'm coming up on 20-minute limits, so I might actually discuss the fluorouracil example in the next video. So we'll see you guys in that video.